Joy Reid knew more about her origins than most people who sit down for a DNA test. A Congolese father, a Guyanese mother. She had names for both sides, countries on the map, a family history with more geographic specificity than the average starting point. The results revealed that knowing where your parents were born and knowing where your family actually comes from are not the same thing. The science went further than the map she had, and what it found there changed the picture entirely. Before we dive in, comment where you are watching from and click subscribe. Her parents came from opposite sides of the Atlantic, and they brought two different worlds into the same household. Her father, Linworth Reid, grew up in the Democratic Republic of Congo before immigrating to the United States. Her mother, Joyce Middleton, came from Guyana on the northeastern coast of South America, a country whose history is Caribbean in character even if its geography is continental. Joy Reid was born in Brooklyn in 1969, the product of that crossing, and she grew up with something most African Americans do not have, a partial map. The map had edges though. Knowing that her father was Congolese taught her something about Central Africa, about a region of extraordinary ethnic diversity, home to hundreds of distinct peoples with languages and histories that predate European contact by millennia. But knowing the country is not the same as knowing the community, the ethnic group, the specific thread of ancestry running through a family before colonialism redrew every boundary on the continent. Congo is vast. Its internal diversity is enormous. The country is a colonial construct laid over a landscape of peoples who had been living in complex relationships with each other for centuries before any European drew a line around them. Her mother's Guyanese heritage presented a different kind of complexity. Guyana's history is a layered thing. Indigenous peoples, Dutch and British colonizers, enslaved Africans brought across the Atlantic, and later waves of indentured laborers from South Asia, all of them compressed into a small coastal country and left to produce a population whose genetic composition can surprise even the people who carry it. A Guyanese family tree does not always look like what the family expects when it is finally examined. Reid went on to build a career around understanding exactly these kinds of historical forces, the long consequences of colonialism, the structural legacies of the Atlantic slave trade, the ways that power shapes what gets remembered and what gets lost. That professional knowledge made her an unusual participant in a DNA ancestry investigation. She understood the history she was being placed inside. What she did not yet know was precisely where inside it she stood. The Congo Basin is the second largest tropical rainforest on Earth and one of the oldest continuously inhabited regions in human history. The peoples who live there, and who lived there long before any outside power arrived to name or claim the territory, represent some of the deepest branches of the human genetic tree. Linguistically, culturally, and genetically, Central Africa is not a single place. It is a mosaic of distinct populations whose differences are as significant, in many cases, as the differences between peoples separated by entire oceans. When the Atlantic slave trade reached Central Africa, it did so primarily through the Kingdom of Congo and the surrounding coastal regions, a sophisticated political entity that had its own diplomatic relationships, trade networks, and internal hierarchies long before Portuguese ships appeared on the horizon in the late 15th century. The trade that developed between European slavers and the Kingdom of Congo began as something closer to negotiation and became, over the following 2 centuries, something far more coercive. By the time the trade reached its peak volume in the 18th century, Central African peoples, Bakongo, Mbundu, and others, represented a substantial portion of the enslaved population carried to the Americas. Knowing that Reid's father came from the Democratic Republic of Congo placed her paternal ancestry inside that broad geographic and historical frame. It did not answer the more specific question of which communities within that frame her family descended from, which ethnic identity they carried, which of the dozens of distinct peoples living within the colonial borders of the modern country her particular line traced back to. Guyana added its own layer of unresolved complexity. The country's African ancestry arrived through the British and Dutch slave trade operating out of West and Central Africa, but the specific ethnic origins of enslaved Guyanese populations are, in most cases, as difficult to trace through documents as any other Caribbean or American ancestry. The families that were brought to Guyana carried their origins with them in memory and practice, and those memories eroded across generations in exactly the ways that memories erode when the conditions for preserving them are systematically removed. The partial map Reid had was accurate as far as it went. The question was how far the DNA could take it from there. Finding your roots is not a celebrity vanity project dressed up as genealogy. The distinction matters because the results it produces are only as credible as the method behind them, and the method Gates's team uses is substantially more rigorous than what most people mean when they talk about DNA ancestry testing. The series functions as a research project with a production budget, archival documentary investigation running in parallel with multi-layered genetic analysis, each line of inquiry checking and extending the other. What makes the genetic side of the investigation particularly significant for a subject like Reid is the depth of the African reference databases the team works against. Consumer ancestry tests have improved considerably over the past decade, but their African population data remains thinner than their European data, a reflection of where the commercial market for ancestry testing has historically been concentrated. Gates's project invested specifically in building a richer African genetic database, drawing on samples from populations across sub-Saharan Africa to allow comparisons that a standard commercial test would flatten into broad regional estimates. For Reid, that depth mattered on both sides of her heritage. Her father's Congolese ancestry required a database capable of distinguishing between Central African populations at the level of specific ethnic communities, rather than simply confirming African origin. Her mother's Guyanese ancestry required a database that could untangle the African, European, and potentially South Asian and indigenous strands that Guyanese genealogy can contain, which makes the analysis more complex than a straightforward African American or West African ancestry analysis. The methodology layered three forms of genetic evidence. Autosomal DNA produced a broad ethnic composition picture across all lines. Mitochondrial DNA traced the direct maternal line to its ancient origin, unchanged across generations. Y chromosome analysis, where applicable, extended the same precision to the paternal line. What the method could not guarantee, and what Gates has never claimed it can, is certainty at the level of individual genealogical descent. Population genetics works with probabilities and statistical similarities, not confirmed family trees. The DNA points. It does not certify. That distinction would matter for at least one finding in Reid's results, in ways that open more questions than it closed. The African ancestry results came back confirming what Reid's family history had suggested, and then went considerably further than it had. Her father's Congolese line produced genetic affiliations consistent with Central African populations, communities from the Congo Basin, whose history stretched back through the pre-colonial period into a deep African past that the modern country's borders do not adequately represent. The specific ethnic affiliations the analysis identified placed her paternal ancestry inside communities that were present in Central Africa long before the Kingdom of Congo rose to regional prominence, long before Portuguese contact, long before any of the forces that would eventually carry Central African peoples across the Atlantic had arrived on the continent. That depth was the first thing the results added that the family history had not contained. Knowing her father was Congolese told her something about modern geography. The DNA told her something about ancient people, about the specific communities whose descendants still live in the Congo Basin today, and whose genetic signature Reid carries in her cells. Her mother's line told a different story, routed through the Atlantic, The African ancestry embedded in her Guyanese heritage traced to West and Central African populations, the communities whose members were carried across the ocean to work on British and Dutch colonial plantations beginning in the 17th century. The specific population affiliations identified in that strand connected her to peoples from the coastal and interior regions of West Africa, communities whose presence in the Caribbean is the direct result of one of the most documented and most violent forced migrations in human history. Two African ancestry strands, arriving through two completely separate historical pathways. One never left the continent. One was taken from it. Both ended up in the same person, sitting in a studio in 2014, hearing the results read back to her for the first time. The confluence that the autosomal results describe was not a simple addition of two known quantities. It was a convergence of histories that had been separated by the Atlantic for centuries before producing a family in Brooklyn. And the picture was not finished yet. European ancestry results required the least explanation and carried the most historical weight. A portion of Reid's genome traced to European origins. The percentage was substantial enough to constitute a genuine genetic presence rather than a statistical trace, and the historical mechanisms that produced it were not difficult to identify. Guyana was a British and Dutch colony for more than 2 centuries. The plantation system that operated there ran on the same logic as the plantation systems of the American South and the broader Caribbean. Enslaved African women had no legal standing, no recourse, and no protection from the men who owned them. The European DNA present in Guyanese ancestry today is, in the majority of cases, a direct biological record of that. Reid knew this. She has spent her professional life documenting the structural legacies of exactly these systems, the ways that colonial power reproduced itself through violence that was legal, routine, and deliberately unacknowledged. Receiving a laboratory confirmation of what the history predicted did not produce shock. Instead, it produced something closer to recognition, the particular feeling of seeing something you already understood abstractly made concrete in a form that is personal rather than historical. What the results could not produce was a name. The European ancestors in her genome left no paper trail connecting to her family, no plantation record, no colonial census entry, no document that closes the distance between a percentage on a chart and a specific person who lived a specific life. The DNA confirmed the encounter, but it could not identify the parties. It could not tell her whether anyone descended from that European line had ever looked back across that history, or whether the silence on the other side had simply continued, generation after generation, to the present. That silence is not unique to Reid. It is a structural feature of Caribbean and American genealogy for millions of families whose ancestry crosses the same colonial fault line. The DNA makes the crossing visible. The people who made it remain almost universally without faces or names. What the science had not yet finished saying was still ahead. Guyana's history contains a genetic layer that most people outside the Caribbean have little reason to know about, and that most people inside it carry without having examined. When British colonial authorities abolished slavery in British Guyana in 1834, the plantation system did not end. It adapted. The solution the colonial government and plantation owners arrived at was indenture, the recruitment of laborers from the Indian subcontinent under contracts that were, in practice, difficult to distinguish from the system they had nominally replaced. Between 1838 and 1917, more than 200,000 people from India were brought to British Guiana under this arrangement. Their descendants today constitute roughly 40% of Guyana's population. The question for Reid's analysis was whether that history had left a trace in her genome. It was a question that only arose because of where her mother's family came from. No other ancestry in the script series would have prompted it. It was specific to Guyana, specific to the particular colonial history of that particular country, and it represented exactly the kind of finding that a DNA investigation surfaces when it is applied to a population whose complexity the family's own narrative has never fully reckoned with. The results showed no significant South Asian ancestry in Reid's profile. The Indian indenture history that shaped Guyana's demography had not, in her particular family line, produced a detectable genetic contribution. That absence was not a surprise in any statistical sense. The mixing between Afro-Guyanese and Indo-Guyanese communities, while it occurred, was constrained by social and economic barriers that limited its scale. Most Guyanese families carry one strand or the other. Reid's carried the African one. What the absence confirmed was the specificity of the investigation. The DNA was not producing generic results. It was engaging the particular history of a particular place and returning findings that were accurate to that history's complexity. The broad picture was becoming clear. What remained was the deepest layer of the analysis, the one that reached past colonial history, past the Atlantic trade, past every documented event, into the ancient origin of the maternal line itself. Mitochondrial DNA operates on a timescale that the rest of the ancestry analysis does not reach. The autosomal results, the ethnic percentages, the population affiliations, and the European colonial thread describe a genetic composite assembled across the last several centuries. They are historically rich and personally significant, but they are recent in the deepest sense of the word. The mitochondrial line goes somewhere else entirely. It passes from mother to daughter without alteration, carrying a signature established in a specific woman at a specific moment in human prehistory and transmitted forward in unbroken sequence ever since. It does not blend with paternal contributions. It does not dilute across generations. It holds its shape across thousands of years in a way that no other form of genetic evidence does, which is what makes it the most precise tool available for tracing the direct maternal line back to its ancient origin. Reid's mitochondrial haplogroup placed her direct maternal line, her mother's line, running back through Guyana, back through the Atlantic crossing, back through wherever in Africa her maternal ancestors originated before the trade carried their descendants to the Caribbean, within a lineage rooted in West and Central Africa. The specific haplogroup branch identified traced to populations that have been present in that region of the continent for thousands of years, not centuries, thousands of years, before the Atlantic trade, before European contact, before the Kingdom of Congo, before any of the political and colonial structures that the rest of the results have been organized around. What that means for the picture the DNA had assembled is this: the Guyanese identity Reid inherited from her mother, the Caribbean ancestry that represents one of the two known pillars of her heritage, traces at its deepest maternal root to the same continent her father's family never left. The Atlantic crossing that brought her mother's ancestors to Guyana was a disruption, a violent interruption of a lineage that had been continuous in Africa for millennia before it was carried away. The DNA had now traced that lineage back past the interruption. What remained was the question of what Reid made of a picture that had grown considerably larger than the map she brought into the room. The map Reid brought into the investigation had 2 points on it: a father from Congo, a mother from Guyana. That was more than most people start with, and she had always known it, had always understood that her heritage gave her a more specific connection to the African diaspora than the blank space most African Americans carry. What the DNA did was take those 2 points and show what lay beneath them, behind them, and running through them in directions the map had never indicated. The Central African ethnic affiliations her father's line produced were not just a confirmation of Congolese origin. They were a connection to specific peoples whose histories predate the modern country, the colonial period, and the slave trade that eventually carried some of their kin across the ocean. Those communities still exist. Their languages are still spoken. The genetic thread connecting Reid to them is ancient and direct. The African ancestry recovered through her mother's Guyanese line added a second connection, routed differently and arriving through the Atlantic crossing rather than through an unbroken continental lineage, but pointing back toward the same broad origin. West and Central African peoples were carried to the Caribbean, their specific ethnic identities eroded by the crossing, but partially recoverable through a science that was not available to them. This was a new kind of recovery. The European colonial thread required no elaborate interpretation. The history of British Guyana explained it. The specific people behind that presence remained, as they almost always do, without names or faces. The maternal haplogroup traced both known halves of her heritage, the Congolese and the Guyanese, back to a single ancient African source, dissolving the geographic distance between them at the level of the deepest available genetic evidence. Haplogroup here maps the oldest part of the story. She came in with a partial map and left with something closer to a full one. Not complete, the names are still missing, the specific individuals still unrecoverable, the European side still silent, but the borders of the map have been extended in every direction. For someone who has spent a career documenting the forces that drew those borders in the first place, the experience of having them redrawn by a laboratory result carried a weight that went beyond the personal. The history she had always studied had, it turned out, been studying her back. What the DNA assembled was a record that her family history had never been able to produce. Central African ancestry reaching back through pre-colonial peoples whose communities still exist. West and Central African populations recovered through a Caribbean line the slave trade had obscured. A European presence whose origins the colonial history of British Guyana explains without requiring elaboration. A maternal haplogroup tracing everything back to an ancient African source. The specific individuals remain unrecoverable, but Reid has spent her career arguing that the forces which erased them were real, deliberate, and consequential. The DNA proves she was right.